So my name is Sandeep Karki. Actually, I'm a fourth year PhD student here at School of Vet. Originally, I come from Nepal, a small landlocked country. It's sandwiched between China and India. Nepal is known for its mountains and its breathtaking landscape. Talking about my background, I am a pharmacist by training. So I did my bachelor in pharmacy from Nepal, and it was from Buccal University, where I stayed for 4 years, another beautiful city in Nepal. I was awarded the scholarship where I pursued my master in industrial pharmacy in Korea. I was always curious to know about how the dog walks inside the human body, the pharmacological response that we generally see once the drug enters the body, and also was very fascinated how a simple drug could be formulated in so many ways. I also have a bit of exposure working in a pharmaceutical industry. After my B.Pharm, I worked briefly for about a year and a half in a pharmaceutical industry called Quest Pharmaceuticals in Nepal, where I got this opportunity to work with different solid dosage forms. I worked with conventional tablets, the capsules with solutions, all this semi-solid dosage form. And after that, when I was in Korea, after my master's study, I took up this job in a pharmaceutical company where I was working with the films. So the projects that's quite similar with what I'm doing in my PhD. So there I was developing different kinds of oral films. So it's one of the alternative for the tablets because it dissolves very quickly in the mouth and it induces pharmacological response instantly. So that's the whole idea behind why you formulate anything in film form. And I also work in long-acting injectables, especially the formulation scientists all around the world. They want the drug to be released very slow for the prolonged period of time. So whenever you are administering any drug, it could be in the body for months, even for 6 months, even for a year. Based on that, you just cut down on the administration frequency of any drug. You don't have to administer many times. So that's the concept of why people are working with the LAI. And when I was in Korea, I was very interested in finding a PhD which is more related with the formulations and the drug delivery, especially the peptide. Peptides are the gold standard these days because every researcher, they just want to work with the oral delivery of peptides. So I was also very interested in working in that project. So I came across this project that was advertised by Professor David Brayden, so I couldn't stop myself. So my PhD is on developing the oromucosal films. That means administering any dosage forms either by buccal, like the inner cheek, or under the tongue roots for the peptides. Big peptides, they are one of the class of therapeutics that's being used widely for the treatment of the chronic and the immune disease. But the majority of the peptides, they are only in the form of injection. You have to administer intravenously, subcutaneously, and there are only few peptides that has recently been launched, and one of them GLP-1 Ribosys, which was developed by the Novo Nordisk, and that was oral peptide. So right now we are working on developing the buccal or sublingual form, the filling of the same peptide form. So we think that developing in the form of peptides, that would be very advantageous, because when you take any tablets, it has to go into the GI tract, then it would get deactivated by the enzyme, and then in the liver there is first pass metabolism, and the absorption is also very low in the guts or the intestine. To avoid all these drawbacks of the oral peptides, we were trying to explore the oromucosal route, especially the sublingual routes, because where the blood vessels are plenty enriched with blood vessels, where you could expect the absorption just to take place instantly. So that's the whole point why we want to develop the pads. And pads, it's very simple because you could not expect any peptide to pass over the biological membrane so easily. So for that, we are integrating lots of enhancers in our formulations. So for now, we are using bile salt. That is one kind of enhancers that has been widely used for the administration of peptide through the oromucosal route. We are also open and been exploring all different options that we could, other classes of formulation enhancers. And talking about progress that we have made so far, because at first we were working for the buccal pep, and buccal pep, from its name, it sounds like through the buccal route, through the cheek route. But the result was not that convincing, the in vivo results that we got from the buccal route. However, we also had plan B, where we were exploring the sublingual route. Sublingual route showed tremendous potential, and you just put that for short period of time, and then you expect to reach the blood level that's good enough to show the response any diabetic, any obese patients. It's available in injectable form, and oral form, but the main drawback with the oral form of the GLP-1, it's absorbed in very low amount. It's less than 1% bioavailability. So with the results that we got in pigs recently, it was almost 19 times more potent what we have seen, but that was just in the big animal study. So later on it has to be translated on the human PK study, other human trials. But with whatever we have achieved so far, things are looking brilliant. So right now the good thing is we got all of our technology patent. We are also looking for the opportunity to commercialize our pads. Not like you worked at the lab scale, so you have to go for the commercial manufacturing. And for that we are working with the Rx Pharma, Novo Nordisk and the Labtex. Actually they are the industrial partners and they've been of great help giving their consultation. They are giving their feedback and even did the scale up at Labtex Germany, where we were able to successfully do this manufacturing of the batch size of 50 grams. That's huge at this stage, but we are planning to expand the pilot manufacturing. That's scaling up to higher scale than what we are doing, and we ended up going for the manufacturing 100,000 of grams. So that would be the batch size. The tricky part, the bottleneck of any formulations to come into the market, it has to get approved. It has to pass through the FDA approval or the EMA approval. So we are working with something that's already been approved, semaglutide. That's the drug. So for us, we have to find the pathway for the innovative medicine. It's not a new drug, but it's already been in the market. So for that also, yes, there is the regulatory pathway. We have to go through the human PK study. We have to do some bridging study. We have to probably also need to do some human trials. We are green signal before our drug gets approved by the FDA or the EMA. It's a process not as comparable with the new drug molecule, but that you are avoiding all the preclinical study, because all this efficacy or the toxicity is well proven in the animals and the humans. So you reducing many experiments that one probably would do. There is the regulatory requirement. We are working with the big animals. We could not directly go to the human as you could expect, because the regulator would not allow that. So for now we are working with a charity based in Berlin. Actually they providing to do the in vivo study. We have conducted many in vivo trials and many in vivo PK study, and recently with our prototype we did 3 or 4 different clinical study with the animals, and all the results were quite comparable. They are very reproducible with what we achieved in the previous trials, and right now we are working on how we could reduce the administration time. So we started off administering the pads for 4 hours, then lately we reduced to 2 hours. Now there is another experiment that taking place next week. For that it would be 30 minutes and 1 hour. So we just want to see if we are able to reach the blood levels just by putting the film for 30 minutes. That means the patients would not have to put the pads for more than 30 minutes. So that's what we are working on. So finger crossed. We have not yet get the result yet, but with what we have seen with 2 and 4 hours, so we are quite hopeful. I think we should achieve the blood levels.